involved. There are a lot of automation there. So if they produce 500,000 more instead of 1 million, who will find out? So again, there are a lot of malicious activity that could happen: cloning, IP cloning, counterfeiting, inserting hardware Trojan. But that video essentially explained just one example, which was malicious activity, putting hardware Trojan.
Okay, very good. I know Professor Humayun will be around this afternoon. You can find him in person, ask a few more questions. Let's thank our speaker one more time.
Thank you.
Just before we break for coffee or drinks, the next part of the program starts at 4 o'clock, and I just want to talk through that. At 4 o'clock, from 4 to 5, we have 4 mini-symposiums running in parallel. We have one on smart health that will be in this room, and then there's a second one just out the doors over there to your right, is the one on RF and millimeter wave technology for communication and sensing. And then there's a third one, a GunRock, which is high-performance programmable graph analytics for a GPU. And then there's one on wireless networking for cyber AI. I just want to make people aware that obviously in this room we have a lot of space. But in the other smaller rooms, we are space limited. The one in RF has about 45, capacity of 45. And then the other two, GunRock and the one on the wireless networking, only have 20 people per room. So upon registration, you were selecting which room to go to. If you selected one of the smaller rooms, you will have preference to go into those rooms. And then if they're not full up, then please, other people can join there. But unfortunately, we have that restriction that we're operating under. But let's break for some drinks and just relaxation, and then we start at 4 o'clock, and then at 5 o'clock, we all come back to this room again, and we will talk about the next part of the program then. Thank you.
Can I have the clicker to walk through? Thank you much. So if you followed all the news let's see if I can get this to work. There we go. So if you follow the development in the space of health, you know that the 4 Ps of healthcare used to be or still are the patient, the physician, the provider, and the payer. And the healthcare is going through this transformation where the 4 Ps for the quality care are going to be predictive care, proactive care, personalized, and preventive. And we think the technology has a lot to do in this space. It's obviously not the only player. There's other components to it. But a lot of it is, on the technology side, is things that the components that electrical and computer engineers can contribute to, whether it's from imaging or data acquisition or data processing. So in the interest of time, we have put together a rather cramped agenda. The first half hour of the session will be a guest speaker delivered by Dr. Stephen O'Driscoll from Verily Life Sciences subsidiary. And the latter part of the session will be a snapshot of some research going on in the department that bridges the gap between some component of electrical engineering and smart health or biology. And I want to emphasize that these are samples. There's by no means, I mean that's an inclusive or a comprehensive coverage of what we do in the department in this space. So with that, I'd like to invite Dr. O'Driscoll to deliver his presentation. As a matter of introduction, Dr. O'Driscoll received his PhD from Stanford in 2011. He was on the faculty at Davis ECE for 2.5 years, during which time he did a lot of wonderful things, received a lot of prestigious awards, including NSF Career Award, and then joined as a member of the founding team of in 2013, I believe, right? Verily, as a member of the founding team of the wearable contactless for glucose sensing, which I'm sure most of you have heard on the news. And he's gone on to be the head of the electrical engineering at Verily, and we're happy to have him here. Please join me in welcoming Stephen.
First of all, thanks to TI for hosting this event. I'm really excited to be here. So this will be a really quick overview of some of the projects in my group. And I start with the group. Now we are in the third generation of students, and actually 2 of them are here. I'm proud of them, Pallavi and Zidong. They carried out a lot of the projects that I'm going to describe here. Our group is very interdisciplinary. We have collaborations with quite a few partners in veterinary medicine, some in medical school, and food science and technology. And we have a few undergraduate students in our group. Just to outline the work in my group, we work with porous metals, and we apply them to biological systems. So one thing I should emphasize here is Stephen talked a lot about circuits element. What we focus on is the sensors interface with the tissues and biologicals. So that's one of the important factors for the sensors and actuators. We use porous metals. We apply them for nucleic acid sensors for detecting pathogens and biomarkers. We also develop neural interfaces for recording electrical activity and modulating neural activity. And recently, we've been combining these technologies to engineer tissue chips for studying more physiological phenomena that cannot be studied in vivo due to convoluting factors. So let's start with the material of choice. This is nanoporous gold. My PhD was on studying mechanical properties of this material. This is basically white gold, a mixture of gold and silver. You leach the silver out, and gold self-assembles into this porous sponge. If you heat it up, you can coarsen it, but preserve self-similar morphology. So it's a really nice material for studying structure-property relationships. It's microfabrication compatible. You can use standard photolithography for integrating into devices. You can functionalize the surfaces with very well-defined chemistry, thiol chemistry, and it's electrically conductive, so it's a useful material for electrical applications. The first one I'm going to mention is the neural interfaces. If you are not familiar with these devices, these are basically these thorn-like devices that are inserted into the brain, and the motivation here is to record the electrical activity and modulate it for rehabilitation. For example, if there's a case of paralysis, you want to bypass the severed region and maybe use it for rehabilitation of the prosthesis or the stimulation of the muscles to regain movement. In a nutshell, can I go back like this? Oh, yeah. So in a nutshell, you have these tiny silicon shanks. These can be also flexible. And you have these electrode sites that basically interface with the neural tissue. One thing that you want to have is a small footprint, just like digital photography. If you have small pixels, you can have higher spatial resolution in the electrical recordings. But you also want low electrical impedance for better matching, so higher signal-to-noise ratio. The problem is when you make these sites smaller, the electrical impedance goes up and basically the signals are buried in noise. Another problem is when you implant these, it's just like if you would get a splinter in your finger, there is a tissue growth around it to protect the body from it, and this basically pushes the neurons away, as you can kind of see here. And neurons are the cells that basically provide the electrical activity that we want to record. So you want to minimize this adverse tissue response and have neurons closer to the electrodes while keeping these scar-forming cells away from it. I'm not going to go into any data, but I'll be around so I can talk a little bit more about it. But what we are doing is we are developing engineering these electrode sites that interface with the tissue via various technologies. One of them is to change the topography so those neural cells can attach to those surfaces while the astrocytes cannot stick to them. We can load these porous structures with drug molecules, so you can release drug molecules and temper that adverse tissue response. And you can also electrically actuate the drug release by electrostatics, and you can push the drug molecules around. So what we are studying now is to record the electrical activity, which could be an oncoming seizure, and then the release drug molecules to suppress that seizure. So a closed-loop control of epileptic seizures. The next thrust area is the biomarker detection. This is of utmost importance in food safety and health, and also plant health in agriculture. So there's need for sensitive and fast methods to detect biomarkers, basically biomolecules in complex fluids like blood or food or plant lysates. And to that end, we've been focusing on nucleic acid-based sensors. So this is kind of a summary slide that shows basically a kind of a cross-section of a nanoporous gold electrode. The idea here is that you have these capture DNA molecules. So if you get let's say a plant lysate or blood, and you would have, for example, DNA or RNA from a pathogen, you chop it up, and then if that chopped piece is complementary to this strand that you have here, you get an electrical signal or a change in electrical signal. So with this, you could actually have very specific information about whether that pathogen is around. A big problem with anything that comes into contact with biological samples is biofouling. So if you have, for example, this electrode working really well, but if you put it in blood, all the proteins basically absorb onto the surface, and it basically occludes the pores, and you wouldn't be able to basically, let's say, it occludes the surface, so you wouldn't be able to detect the DNA. But this matrix structure actually helps with that, where the large molecules are stuck on the surface, but those small nucleic acids can still go into the pores. And with this one, you can still contain, basically detect the electrical activity. How much time do I have? 1 more minute? Or? Okay. So what we are doing is we are basically combining these technologies to develop these platforms where we can, for example, have brain tissues in 2 chambers, and we are studying how inflammation propagates from one tissue to the other one. And we are recently looking into how we can study the gut microbiota. You may have heard about these. Basically, the microbiota health can influence a lot of the health conditions. So basically, we are using this recording technology and these tissue chips to get electrical signatures of the microbiota, so we can, in high throughput, identify what factors lead to what type of disorders. And finally, one last thing we are focusing on is developing video games for basically teaching microfabrication. So one of the undergrads developed this clean room genie that gives you instructions. So with that, this is just like a super quick overview. Thank you, and catch me in the reception if you have any questions.
Thank you. So time for a quick question while we're getting the next speaker set up, if there is one.
Here you go, Cheung. So gold for us, the thing with the gold is the coarse gold is the controllable morphology, which gives you a lot of different functions. Okay. There's platinum black, which is more of a spiky structure. Titanium is not as good for forming the electrode, but it's good for mechanical. I can describe it.
So next we have Professor Weijian Yang, who was also recognized earlier in the program as a recent awardee of NSF Career Award.
Great. Thank you very much for the introduction. So it's my great honor here to be discussing some of our research entitled of optical methods in brain research. So what we want to do is actually we want to read information from the brain and also write information into the brain in cellular resolution. And then by doing this, we want to investigate how the neural network works, and then whether we can prevent some of the brain disease. So what we are seeing here is actually the cellular resolution brain imaging. Whenever you see that there's some blinking, that means that neuron has some activity. Probably that's what we are doing as well in our brain. This is for the in vivo image on a brain, on a mouse visual cortex. So our goal is that during the animal's behavior, we want to imagine the brain in cellular resolution over a relatively large spatial domain. And then on top of that, we can do using some machine learning to map out what's happening on this neural network. And then after that, perhaps we can use optical techniques to manipulate individual neurons' activity. So we essentially write information into the brain. And then by doing this, we may be able to map out the brain connectivity, or maybe to inject some memory into the brain, or maybe you can predict the animal's behavior. So by doing this, we actually close the loop. So this is about the optical method. So in reality, there's also other technology to study the brain. So the most prominent one is actually electrophysiology. So it can do a few cells recording and stimulation at a time. But however, its field of view is pretty small. Usually, or maybe we can use electroarray, but that is relatively invasive. Though with Stephen's work and also Erkin's work, we might be able to miniaturize those devices to make it less invasive. But that is under development at this moment. And the other spectrum is we can do microscale, basically using fMRI, beautiful mapping of the whole brain. But the problem is that the spatial temporal resolution is relatively poor. So what we want to do is that we are trying to use optical methods to investigate the mesoscale. That is essentially the bridge between microscale and macroscale. And then within this mesoscale, we can investigate how the neural network is working within like probably 1 millimeter by 1 millimeter field of view. And then we can have cellular resolution, also reasonable temporal resolution. So now our research is trying to increase the optical throughput as well as the accuracy of the photostimulation to manipulate the brain activity. So this is one of our work. What we are doing here is that we are using two-photon microscopy to rapidly scan multiple layers of the brain tissue in vivo. And then on top of that, we use holographic approach to photostimulate each individual's neurons. Whatever group of neurons that you watch, I make a hologram, I photostimulate those. So in that case, if we know which group of cells that we want to stimulate, we might be able to directly inject the information into the brain. So on the right-hand side, is showing that by using a holographic approach, we can indeed project different patterns into the brain. So this is kind of like a cartoon showing that we can do 3D holographic. Imagine that each of them is a neuron that you can photostimulate that. Once it's photostimulated, that neuron will have activity. So you are essentially writing information into the brain. So using this technology, we are actually studying some interesting biological questions. So one of them is that whether we can reprogram the brain. Just using light. So this work is based on the Hebbian's theory, saying that if the neurons fire together, they will wire together. Once they wire together, that means that they actually form a group. Optogenetics it's actually the photostimulation. It can we try to use that to artificially implant the neural ensemble. So within the next 1 minute, I would like to show you how we are doing this. So this is like a neural network in the brain. Each of them is individual neurons. And then some of them is closely connected. For example, these three. When this cell have activity, the other two will also have activity because they form a group originally. Now, what we do is that we use photostimulations. So we photostimulate a group of neurons with cellular resolutions, and then we photostimulate that for a great amount of time, about 30 minutes. And then as a result, this group of neurons will have continuous co-activation. And then now following this, if the neurons fire together, they will wire together. So what we are doing here is that during the photostimulations, we actually grow the underneath connectivity between them. So their connectivity becomes stronger. And then now finally, after we finish this, then we actually find that this group of cells will have stronger connectivity. Their spontaneous activation becomes stronger. So in this case, we actually artificially inject some information into the brain, and this can actually last for a few days. So we plan that perhaps in the future, we can reprogram the memory, for example. And then now, in addition to this work, we are also working on some other domains. For example, the photonic device and systems. We are actually making implantable device, miniaturize them, so that we can put into the animals' heads or for biomedical application and neural activity recording and manipulation. And then finally, we want to use these tools to study how the neural network is actually working, and particularly how memory can happen and how learning can happen. And I believe that using optogenetics and photostimulation, it's a very nice tool. And then because we are in the engineering department, so my other research area is that trying to learn from biology and then using the bio-inspired informations to make engineering better. So this is our funding source, and thank you very much.
Any question while we switch to the next speaker? Which happens to be me.
right? So if not, then we can probably move on. Yes. I'm still here, so after that we can talk.
So I guess next we have Dr. Cheung or Gregory Rehn to present from Cheney's group. Please.
Is it just this? Is this the clicker? Okay, thanks. Hi, my name is Gregory Rehn. I'm a PhD student in Dr. Chua's lab. Dr. Chua couldn't be able to be here today to present, so I'm just coming in her stead. So basically, we have a number of different projects that we're working on in the lab. We partner with a bunch of different doctors over at the UC Davis Medical Center. And the area of research that I'm actually doing is working on the ventilator. So one of the things that we want to do with the ventilator is we want to kind of find it and use different data types than we use traditionally in the hospital. So one of these is like the traditional lab values that we get just from drawing blood or fluids from a patient, or maybe a chest x-ray. So, and then we've been developing a number of different devices. So one of the devices that we have right now is called Pac-Man. Pac-Man is basically an automated resuscitation and fluid monitoring device. And then, so basically what I'm going to do in my talk today is I'm just going to talk about the project that I've been working most closely on, which is basically performing detection of this disease called acute respiratory distress syndrome. And as you can see from the text, it's an extremely serious disease. It occurs very frequently in the intensive care unit, and it's very deadly as well. But we've also been working on a number of different diseases as well, including, as I mentioned, adapted fluid response for developing these devices in Pac-Man. And we've also been developing new screening algorithms for congenital heart disease as well in infants. So at least just the way that we've been collecting data from the ventilators is we attach a Raspberry Pi to the ventilator, and then we collect that data and then send it over to a server. Doctors can actually utilize that information by, through an app and view what the patient is doing in real time. So this is just kind of an example of what our data stream looks like, and then we can actually start analyzing that information using Python encoded algorithms. And so just looking at this in real time, we can get this ventilation information, and a couple of the advantages are is that this ventilation information is always happening as the patient is hooked up to the ventilator. And so it's ubiquitous, continuous, objective. Objective because it's being read by a machine. And then what we can do after that is if we want to use traditional machine learning algorithms, we can extract information from these things. So we just call this physiologic information. And so a couple examples of that would be the amount of air that a patient is breathing in for every single breath. And then what we can do after that is we can just basically do analytics on that. And then one of the diseases that I've been working on detecting is called ARDS, as I mentioned before, the acute respiratory distress syndrome. So we want to improve the time to diagnosis for this because doctors already have a lot of difficulty diagnosing this to begin with. So we actually started out with utilizing a random forest model to analyze the data that we're getting from the ventilators. And then we've been looking at windows of breaths and basically extracting information from them. And then through this project, we've been able to collect a study cohort of about 100 different patients. If anybody works here in medical devices, I'm sure you know how difficult it is to actually collect study patients. And once we collect all this information from the ventilator, we can make classifications on whether or not this patient is might have ARDS, whether or not they do not look like they have ARDS, or maybe if they have a chronic obstructive disease as well. Yeah, and then we can basically perform a classification on the number of classifications that the machine learning algorithm made by hour, and then possibly we could send a recommendation to the doctor like, hey, look at whether or not this patient has ARDS, just basically based on the fraction of votes that the machine learning algorithm made. Like if, the number of windows classified as ARDS is greater than 55% or 50%, we can just set like a threshold, maybe just send a warning to the doctor. And our results have been pretty good so far. And yeah, I'll leave it to Dr. Cheung.
Did I press something? Okay, good, thank you. My name is Samson Cheung. I'm the John's faculty of the ECE departments. I'm also a faculty at University of Kentucky. So I have been co-leading with Professor Chu on 2 projects that I'm going to tell you something about. The first project is on using machine learning to help early diagnosis of autism. So autism is one of the most prevalent neurodevelopmental disorders in US. CDC estimated that 1 in 59 children in US is in some form of autism. And the key to the recoveries of autism is really early interventions, as early as possible. In US, the average age of diagnosis is 4 years old. However, there are many studies actually shows that parents and clinicians notice the first emergence of the symptoms already started around 6 months old. So we are losing about 3.5 years. And the goal of that is that we try to identify behavioral markers from, for example, parents collected videos in order to push the diagnosis age as early as possible. So our partner in crime is Dr. Sally Osnoff of the Mind Institute at UC Davis. She is a renowned autism researcher. In the past 15 years, she has been conducting, wrong direction. The green one? Oh, of course. So she has been conducting one of the largest sort of the sibling studies. Basically, is that she recruits children whose at least one of the siblings have autism. Okay, that has basically get a much higher propensities for the child to have autism, and basically collecting videos from 6 months old all the way track to 36 months. So it's a really kind of a long longitudinal studies that's really actually is perspective because it started collecting data before the emergence of the symptoms. And so she has collected one of the largest video data sets. That's over 5,000 hours of videos. And actually, they are hand-coded. Hand-coded, she identifies a number of what she and her research team believe to be kind of a clinically significant behaviors, like eye contact, like some social smiles events, and trying to do that to correlate with the onset of autism. So what we are trying to do, and we just got funded by NIH R01 in November last year, is try to do that to automate the process using machine learning. So we have considered right now, we're developing the different neural network techniques to look at the entire videos and using a kind of a myriad of different types of features from facial markers, eye gaze, eye contacts, body pose, in order to detect these types of behavioral markers in order to predict the onsets of autism. Another project that's, again, we sort of recently started, and it's sort of looking at sort of the other ends of the disease that happens later in life, which is Alzheimer's. So Alzheimer's disease occurs, I mean, if you look at the adults in the US, 65 years and older, about 10 to 15% of people have some form of Alzheimer's disease, and this is disproportionately affects people with African Americans and Hispanic origins. And CDC estimates that actually, by the year of 2060, we'll have more than 15 million people with Alzheimer's disease. So it is really important for us to better understand from the molecular levels, cellular levels of what the what Alzheimer's disease contributes. So luckily at UC Davis we have the Alzheimer's Disease Center, Seker, which is kind of one of the largest data collections of brain tissues from peoples who, sort of postmortem brain tissues from people who have Alzheimer's disease and related disorders. And again, our partner in crimes, in this case, Dr. Brittany Duggars, are very interested in different types of pathologies in Alzheimer's disease. More specifically, she is interested in the amyloid beta plaques and cerebral amyloid angiopathy. So basically, beta amyloids are proteins, normally occur proteins in brains. However, for Alzheimer's disease patients, the levels of the beta amyloids are increasing to a toxic level, and they form plaques to block the communications between neurons, or the deposits around the walls of the blood vessels to block the glucose nutrients deliveries to the neurons. So what happens is that the neurons start to die out, and then the brains of the AD patients will shrink significantly, and then affects a lot of communications, thinking, learning, memory functions. So the studies of these types of plaques, especially the distributions around the brain tissues, are very important. And what you notice is that there are disproportionately larger number of these plaques in the gray matters over the white matters. And then, so solving kind of from the brain imagings, solving the segmentation problems of separating gray and white matters becomes important. It used to be done, need to be done manually. So what we're trying to do is to use, again, computer vision and image processing techniques to automate the process of separating gray and white matters. So again, this is a project that started recently. We have got some encouraging results. So there is actually a poster that I think two of the students here who work on those things is actually going to present. So I'm not going to get into any details. So, but make sure that you stay tuned for that. So that's my pet. Okay, thank you.
So thank you for your attention. We're out of time. We actually had more material than what we could present today, but I wanted to, again, repeat the point that there are more posters, and that please enjoy, and you can follow up on some of the research that goes on in the department in this area of smart health. So thank you again for your attention. It's just the time.
I think I started.
Yeah, I know. And in general, we didn't do a good job of organizing it because he was a